Today I'd like to talk about different types of cell division in eukaryotes, but before we can get to that, we really need to get some background information. We're going to be talking about chromosomes. Chromosomes are composed of chromatin, and chromatin is composed of DNA and proteins. So that when we look at the structure of a chromosome, there's a DNA double helix. It wraps around clusters of 8 proteins, histone proteins, forming these structures called nucleosomes. And then nucleosomes can be compacted at certain times and relaxed at certain times so that genes can be accessed and the information in the genes can get out. But we'll talk about some of that down the line. So what I'm interested in right now, or what I want to get into next really, is the structure of DNA. So again, this DNA can be compacted or relaxed depending on the needs of the cell. So DNA is one of 2 types of nucleic acids. There are 2 nucleic acids, DNA, which stands for deoxyribonucleic acid, and ribonucleic acid, which is RNA. And we'll talk more about RNA in future modules. Today we're going to focus primarily on DNA, which is the primary genetic material. So looking at the structure of DNA then, DNA was, its physical structure was determined by James Watson and Francis Crick back in 1953. And in the Watson-Crick model, we have 2 strands of DNA, and they form a double helix. So this is a view of that, of their form of DNA. And the DNA is formed of individual repeating units, which are called nucleotides. And so as we look at a nucleotide is composed of a sugar component, a phosphate, and also a nitrogenous base. And there are 4 possible nitrogenous bases that can be used in this position: adenine, guanine, cytosine, and thymine. So again, as we look at the structure of DNA, there's a sugar phosphate backbone, and then the nitrogenous bases come off perpendicular to that backbone. And you'll notice that one strand runs in one direction and the other strand is literally upside down compared to that. So we have the sugar phosphate backbone here, and the nitrogenous base is coming off. And then the 2 strands are held together by hydrogen bonds between the nitrogenous bases, but I'll get back to that in a second. So again, looking at the structure of an individual nucleotide then, again we have the sugar phosphate backbone with the nitrogenous bases coming off perpendicular to that. And it is really the order of these bases which is significant. And so part of how we determine directionality with DNA is by looking at the sugar component of the nucleotide. Because again, it's the actual order of the nitrogenous bases that really determines their heredity. That's what makes an elk different from a deer and makes them different from you and I. It's not the individual nucleotides, it's the order of the nucleotides that's significant here. And so just as in English, the letters of English can make words. If you read from left to right, that's going to give you a different message than if you read right to left. And so in the chemical world, the real physical world, there's 3 dimensions, things can move around, so we need kind of chemical directions when we read this code or read this language. So in looking at the sugar then, when we look at the sugar, there are different carbons, 5 different carbons here that make up DNA, and those are numbered, carbon 1, 2, 3, 4, and 5. And so when these sugar and phosphates form the backbone of DNA, then we can see the sugar 3, 4, 5, we can see 3, 4, 5, we can see 3, 4, 5, 3, 4, 5. So the last carbon we get to on this sugar would be the 5 carbons. We call this the 5' end. Down here, the first carbon we see is the 3 carbon, so that's the 3' end. So when we talk about the order of bases, then we want to make sure we know whether we're looking and reading 5 to 3 or 3 to 5, because that will determine, what characteristics we find. And again, we're looking at 4 possible nitrogenous bases. Notice that cytosine and thymine have a single ring structure. Guanine and adenine and guanine have a double ring structure. So these are pyrimidines, these are purines. And so it is this nucleotide sequence then that determines the amino acid sequence of a protein. Proteins, that determines a protein shape, which determines a protein's function. And it's the proteins ultimately then that determine the characteristics of an organism. So when we look at the genetic makeup of a chromosome then, chromosomes don't so much code for, say, blue eyes as much as they code for proteins that will make your eyes blue. So again, this nucleotide sequence is the key. And changes in that nucleotide sequence affect protein shape and protein function and physical characteristics. So again, we're looking at two strands complementary to one another, not really identical to one another. So again, we're always going to find adenine and thymine associating. We're always going to find guanine and cytosine associating in this double helix. And so the strands are not identical to one another. We describe them as being complementary. So it's just like your shoes are not actually identical to one another. They're complementary. They fit together, but they're not actually identical. And so when we look at one strand and another, then we're always going to see this association, adenine to thymine and cytosine and guanine. And 3 hydrogen bonds between cytosine and guanine, 2 hydrogen bonds between adenine and thymine. So what's interesting then about DNA is if you know one strand, you also know the other strand because of this association of adenine, guanine, cytosine, and thymine. So if I were to ask you to draw then a short sequence or a short portion of a chromosome, let's say I want you to draw 5' guanine, thymine, adenine. Here we're looking at 3 nucleotides, so you'd start with your sugars, you'd put in your sugars. You'd link the sugars together with phosphate groups. And then again, starting at the 5' end, try to identify that. So again, here's our 5' carbon here, 543, 543, 543. So this would be the 3' end here. So starting at the 5' end, we'd want to put in our guanine. We'd want to put in, which is a double ring structure. We'd want to put in our thymine next, which is a single ring structure. And then we'd want to put in adenine, which is a double ring structure, and you could label those. And then because we always associate guanines with cytosines and thymines with adenines, then if you know one strand, you also know the sequence of the other strand, or you also know what the other strand is composed of. So again, opposite, you'd go ahead and put in your sugars, but they are going to run in the opposite direction. And so opposite, and we'd connect with our phosphates, of course, to create the backbone. And then opposite guanine, you'd end up putting cytosine, which is a single ring structure, opposite thymine, adenine, double ring, opposite adenine, thymine, which is single ring, and then the hydrogen bonds linking those together. So again, this is an important quality of DNA then, and so one strand will be coding for proteins, the other strand basically is just a placeholder. And also what that can do then, if there's damage to one of these nucleotides, maybe something knocks this nucleotide out, there are enzymes that can come in and say, oh, okay, this is adenine, so I need to put a thymine opposite there. So it's really handy for repairing DNA. And again, one strand runs in 3 to 5 in one direction or 5 to 3. This one will run 5 to 3 in the opposite direction. So they are literally upside down relative to one another. And if you know one strand, so this is simplifying it. So here I'm just showing the backbone as a line, looking at a sequence of bases. Then, if you know one strand, you can build in the other strand, because anytime you have guanine, cytosine goes opposite. Anytime you have adenine, thymine goes opposite, and vice versa. And so if you know the sequence of one strand, you know the sequence of the other strand. Okay, now another idea we need to talk about too as we look at cell division then is something called ploidy. Ploidy tells us how many sets of chromosomes a particular cell has. And so some cells in nature have a single set of chromosomes, and that's described as being haploid or 1N. And so when we look at humans, for example, we have 23 different and distinct chromosomes. So the information on chromosome number 1 is different than number 2, different number 3, all the way through 23. And so the only cells in our bodies that would be haploid would be our sex cells, our gametes, sperm or eggs. And so if a cell has a single set of chromosomes, again, we describe that cell as being haploid. And so that's going to be our sex cells. Now, most of the cells in our body are diploid. And if a cell is diploid, what that means is that there are 2 sets of chromosomes. And we describe these pairs of chromosomes as homologous pairs. So in fact, all the cells in our body except for sperm and eggs would be diploid. And so again, we have 2 sets of chromosomes because we get 1 set from mom, we get 1 set from dad in sexual reproduction, and then those join together to form a diploid cell. And so when we look at chromosomes, then we're normally talking about pairs of chromosomes. We have 2 of chromosome number 1, 2 of chromosome number 2, 2 of number 3, all the way through 23. So most organisms in nature then are either diploid or haploid. So diploid organisms produce haploid gametes, and then when those gametes, sex cells, fuse together, that reestablishes the diploid condition in the next generation. But we'll get to that in a few minutes. So that occurs in sexual reproduction. Now, there are a few organisms in nature, mostly plants, that are polyploid. And so polyploid is saying that there are many sets of chromosomes. And again, there can be like literally that. You can be 3N, 4N, 5N, 6N, 8N, 12N, whatever it happens to be. So again, we see this mostly in plants. And an example of a plant from the Yellowstone ecosystem would be Indian paintbrush. Indian paintbrush is polyploid. And what's also kind of interesting is that plants that are polyploid are more likely to be at high altitudes, or also close to the poles. So colder climates tend to bring about polyploidy. Okay, now this brings us then to cell division. And so there's two types of cell division. One type is mitosis. And in mitosis, you're going to start with one cell. Typically, it's a diploid cell. It doesn't have to be, but typically it's diploid. And the chromosomes will be copied. They'll be replicated so that you double the number of chromosomes. And then those will be divided into equal groups, identical groups, and then the cell will divide in two. So you start with one cell, you end up with two cells. There's no change in ploidy here. So again, most of the time we're talking about diploid cells that give rise to diploid cells. And what's important to understand about mitosis is that these daughter cells that are produced are genetically identical to one another, and they're genetically identical to the parent cell. So again, there's no change in ploidy in mitosis. You replicate the chromosomes, double the number of chromosomes, separate them into identical halves, and then divide the cell in two so that you end up with two cells that are genetically identical to one another. Now, the role of mitosis in nature, there are several things that mitosis does. One is it can be used for reproduction. So if you're a single-celled organism, you divide in two, you now have two organisms. That's an example of asexual reproduction. Some animals are capable of asexual reproduction. Sponges, for example, this is something called a hydra, a freshwater organism. Some primitive animals are capable of asexual reproduction. And asexual reproduction means reproduction without sex, without a partner. And so aspens in the Yellowstone ecosystem also, and in plants in general, I would say, can asexually reproduce. So again, they produce new cells. But the important thing to understand about asexual reproduction is that the offspring that are generated are genetically identical to the parent organism. So, as we used to look at a stand of aspens like this and think that these were all produced by seeds of different trees, what we now realize is that they all share the same root system and that all of those trees are genetically identical to one another. So we refer to those actually as a clone of aspens as opposed to a stand of aspens because they're all genetically identical, really part of the same root system. Some people describe them as part of the same organism. So all of that tissue then is generated through mitotic division. Another role of mitosis would be for growth and development. So at one time we were all one fertilized ovum. We were all babies at some point, and so as the young add cells and differentiate and grow, that's how the young get to be adults. And so again, that's part of growth. So growth means you need new cells. New cells are produced through mitotic division. So all of the cells produced through mitosis then are genetically identical to one another, meaning that really all the cells of your body are genetically identical to one another, except for those that you use for sexual reproduction, sperm or eggs. And then still another function for mitosis would be, for tissue repair or tissue renewal. So, if you damage your skin, you get a cut, broken bone, mitosis will generate new cells for the repair that's caused by that damage. So mitosis is part of a larger process. It's what we call the cell cycle. And in the cycle, I think what I'll do is kind of start here with this G sub 0 phase. That's where a cell basically is doing whatever it's supposed to do. A liver cell being a liver cell, kidney cell being a liver cell, kidney cell rather, skin cell being a skin cell. And what initiates mitotic division is what we call the S subphase of interphase. And this G sub 0 phase is part of something called interphase. And the interphase cell is a normal looking cell. When you look at that cell, the nucleus, it looks normal. Nuclear envelope is intact. There's no obvious chromosomes. It just looks like a normal cell. But what initiates actually cell division is what we call the S subphase of interphase. And this is the synthesis phase. This is where DNA is replicated. So in interphase, this S subphase rather of interphase, what happens is that the chromosomes will replicate. And this is accomplished by something called DNA polymerase. So DNA polymerase builds DNA strands, and it matches complementary bases to an existing strand, and the new strands will always be built in the 5 to 3 direction. So that if we're looking at a chromosome, and chromosomes actually are millions of bases long, not just the few that we're showing here, what's going to happen in this S subphase is that the two strands of a chromosome are separated from one another by a series of enzymes, a network of enzymes. And then DNA polymerase will come in, read the existing strands, and build in new strands. So again, any place where there was guanine, cytosine would go. Wherever there was cytosine, guanine would go opposite that. And the new strands are built in the 5-3 direction. So that at the end of the process, then, you end up with two chromosomes that are genetically identical to one another. And each chromosome then has one original strand and one newly constructed strand. So we refer to this as replication rather than just duplication, because you don't take one chromosome and build a new one. You take the original chromosome, split it apart, build in new strands so that each new chromosome is part old and part new. So again, eliminating the base pairs here, but it's the idea is that chromosomes will be separated. They build, the polymerase would be the circles here. Polymerase comes in, starts building. As the chromosome unzips, in one direction, it can be built continuously. In the other direction, it has to be built in pieces. We're not going to worry too much about why that happens, but that's kind of the reality. But at the end of this process, again, we're going to end up with identical chromosomes. So the cell looks normal while this is going on, but again, in the S subphase, chromosome number is doubled. And then those chromosomes are divided into identical parts, and then the cell divides so that we end up with two genetically identical cells. So again, there's a series of phases that we see in mitosis. There are physical changes that occur in the cell. So again, in the interphase cell, the cell looks normal. At the end of the synthesis subphase, the chromosomes have been replicated. And then there's what we call gap phase 2, where the centrosomes are replicated, and basically the cell actually gets ready for the physical division. And so these phases are prophase, metaphase, and anaphase, which are responsible for dividing the chromosomes from one another, and this is known as karyokinesis. And then there's another phase called telophase, and that's where actually the cell is divided into two new cells. So next I want to look at mitosis. What are these phases of mitosis? And we're going to look at an animation here that kind of shows you what I'm talking about so you'll understand this mechanism. So in prophase then, what happens is that something called a mitotic spindle is formed. And so this is actually going to be the machinery that's going to be separating the chromosomes. The nuclear envelope will begin to disintegrate, and also the chromosomes themselves will begin to coil and condense and become visible. So in interphase, you can't see the chromosomes, but in prophase, you begin to see these chromosomes as the mitotic spindle is being produced. And a chromosome and its replicate will be attached to one another by something called a centromere. And so as this mitotic spindle again completes, nuclear envelope disintegrates, the chromosomes become visible, these tubules, microtubules, will attach to the centromeres that are holding the chromosome replicates together. And we refer to these chromosomes and their replicate as sister chromatids. So microtubules will be attaching to these centromeres of the sister chromatids. And then these microtubules will line them up all at the middle of the spindle. We refer to that as metaphase. So there's a lot that goes on in prophase. But in metaphase, they all line up essentially in the middle equator of this spindle. And then they'll be pulled apart in what's known as anaphase. The centromere replicates, and so that allows the two sister chromatids to separate, or the chromosome replicates to separate. They get pulled to either end of the cell. The cell elongates in this process, and then the last step is effectively to divide the cell in two. And we are looking at an animal division here. Same kind of thing happens in plant cells with a few changes. But in telophase, the cell actually divides in two. And so we're left with two daughter cells. Each would be diploid. If we started with a diploid cell, they would be genetically identical to one another. Okay, so again, that's the mechanism by which we start with one cell, end up with two daughter cells. Now, meiosis is another type of cell division, and this is a type of cell division that produces haploid cells, produces the gametes in most organisms. And so meiosis is not sexual reproduction, but it's required for sexual reproduction, because in sexual reproduction we want to get from diploid cells to haploid cells so that those can then join with a partner to reestablish the diploid condition. So meiosis then is going to get us from diploid cells to haploid gametes, sperm or egg. Those will fuse together in fertilization, also called syngamy, and then that results in a diploid zygote, and then through mitotic division, new cells are generated to produce the new individual. So what we're going to be interested in meiosis is actually getting to this haploid condition. How do we get from diploid to haploid? And then also, what are the downstream effects of that? Why is that a good thing? And in sexual reproduction, there's an advantage to sexual reproduction. The disadvantage is you need a mate, you need a partner. You have to find somebody to reproduce, whereas in asexual reproduction, you don't. But what's good about sexual reproduction is that it produces a lot of genetic diversity in the population, and that seems to be a good thing, because when there's less genetic diversity in a species, they tend to have problems, but we'll come back to that later. Okay, so in meiosis then, what happens is we have a single round of chromosomal replication. So you're going to double the number of chromosomes, and then that's going to be followed by 2 rounds of division, so that you end up with not 2 cells, but 4 cells, and these 4 cells will be haploid instead of diploid. And what's really important is that they are genetically dissimilar to one another. They are not genetically identical to one another. So it's really impossible to produce gametes that are identical to one another. So genetic diversity here is generated not only because in sexual reproduction, because you're going to have a partner, but also meiosis itself generates diversity, and we're going to talk briefly about how that happens. So as we look at meiosis then, again, there's kind of 2 phases here. There's what we call meiosis 1, where we go from 1 cell to 2, and then meiosis 2, where we go from the 2 cells to 4 haploid cells. So again, 1 round of chromosomal replication to double the number of chromosomes. Then we first split those into 2 parts and then separate those again, so there's 1 set of chromosomes in each cell. And what makes these gametes then genetically dissimilar to one another is 2 processes. One's crossing over, and another is known as independent assortment. So again, meiosis then will generate the gametes, and then in sexual reproduction, gametes fuse to produce the new individual. That gets us back to the diploid condition. So we inherit 2 sets of chromosomes. We get to pass on 1 set of chromosomes. Our partner passes on 1 set. That gets us back to the diploid condition in the next generation. So again, this is why there's a lot of genetic diversity, and this is why even siblings, will not be genetically identical. There's going to be difference even among siblings, because the genome is scrambled in meiosis, and also you're bringing in a partner. So I'm not going to get into a lot of details of meiosis. Again, we basically use the same kind of descriptions. Again, there's different phases here. Again, meiosis 1 gets us from 1 cell to 2. Meiosis 2 gets us from 2 cells to 4. But at the end of that, we'll have 4 haploid cells. But what I do want to get to is explaining what independent assortment is and crossing over is. So what independent assortment is, again, is that we have 2 sets of chromosomes. We get to pass on 1 set of chromosomes. So there's some options here in terms of what we pass on. We could just pass on 1 set of the chromosomes that Dad gave us. That's one possibility. We could also just pass on 1 set of chromosomes that Mom gave us. That's another possibility. But we could also pass on 1 set of chromosomes that is a mixture of our dad's chromosomes and our mom's chromosomes. And that's what independent assortment is. So in meiosis, and again, we're not going to worry about the details of the process, but there are a couple of stages in meiosis where we scramble our chromosomes so that we're not just passing on 1 set of moms or 1 set of dads, but we're passing on a single set that is a combination of chromosomes that we inherited from mom and dad. Now, crossing over is really a crazy process. Crossing over ensures that we don't even pass on a single chromosome like we inherited. So what happens in crossing over is that first, the chromosomes are replicated, and they'll be attached by centromeres. And then our like chromosomes will associate with one another. We call this a tetrad. So again, we would have a chromosome number 1 of dad, a chromosome number 2 of mom. Those would replicate, and then our mom and dad chromosomes of each type associate, and then there are enzymes that will cut and splice those together so that at the end of crossing over, we no longer have any chromosomes, or in this case I'm showing you we have some that are like what we inherited from our dad, like we have from our mom, but we also have chromosomes that are chimeras, or like a mosaic of what we inherited from mom and dad. So this doesn't produce new traits, but it produces new combinations of traits. So dad might have brown hair, brown eyes. Mom might be blonde hair, blue eyes. Now we could actually have chromosomes where you have blonde hair and brown eyes, or brown hair and blue eyes in combination, whereas previously we didn't have chromosomes like that. So again, then we pass on one set of chromosomes that is not only a mixture of mom and dad's sets of chromosomes, but the individual chromosomes themselves are chimeras, or mixtures of mom's and dad's chromosomes. So it's a really amazing thing. So again, independent assortment says that we will not pass on a complete set of our mom's chromosomes or a complete set of our dad's chromosomes. And crossing over ensures that each individual chromosome is a mixture of mom's and dad's traits. And so I think one kind of an analogy I could use here is let's say that I've inherited a trunk of clothes from my mom, and I've inherited a trunk of clothes from my dad, and I have a child, and I want to pass on one trunk of clothes to my child. So I have some options here. So one thing I could do is I could just give my child a trunk of my mom's clothes. I could give my child a trunk of my dad's clothes. But I could also take some of the clothes of my mom, some of the clothes of my dad, and pass those on to my child and let them keep that as an heirloom. That would be the equivalent of independent assortment. So in other words, I'd take maybe a shirt of dad's, a skirt of my mom. So I'd take a mixture from each trunk and pass those on. That would be independent assortment. Crossing over would be if I decided, well, I don't want to just pass on a shirt of my mom's and a blouse, or a shirt of my dad's and a blouse of my mom's. What I'll do is actually cut up the blouse and the shirt, and I'll put the sleeve from one onto another, and I'll actually make a garment that's a mixture of mom's and dad's garments, and I'll pass on that trunk. So that's what crossing over is. So between independent assortment and crossing over then, what you're doing is scrambling up your own genome, and you're passing on chromosomes unlike anything that you actually inherited. And then when your mate does the same thing, then that produces an extraordinary amount of genetic diversity in the population. And that seems to be a good thing, because that's what most organisms do in nature. All right, see you next time.